So I'll be talking about correlative light electron microscopy, and I'll take you a bit deeper into tissue CLEM. And so what I will do is, first of all, I'll talk about CLEM, correlative light electron microscopy. What do we mean by that? And then I'm going to show you one workflow which I've tentatively called simple CLEM. It's not that simple, but it's on a 2D cell culture, which makes life much easier. And then I'll explain how we have tackled the problem of going into a tissue and finding back structures inside a tissue. So correlative light electron microscopy is really the observation and analysis of one and the same event by both light and electron microscopy. And why would you do that? Well, we all know this is mainly a light microscopy conference, so you all know the benefits of doing live cell imaging. You have, in that case, a history of an event. So if you look at well over 10 cells in this movie here, so you have this wide field of view, you have lots of cells, and you can see moving things around. So that gives you a history of events. If you look here as well, then you have a specific cell that has a blue marker in there. So we can pick out that one cell and look at that. On the other hand, we have electron microscopy. That gives us a much higher resolution, so we can zoom into, for instance, that one particular blue cell, but it also gives us a reference space, because what it does, in light microscopy, we have to label. Most of the times, we've seen a couple of examples earlier that you don't need to use fluorescence, but most of the time we use fluorescence, so you have to know what you're looking for. In electron microscopy, we get that as an added bonus. So we can see the membranes that are bound, for instance, for the endocytic vesicles. We can see the cytoskeleton and those kinds of things we get as a bonus. So that reference space is really quite critical for electron microscopy. Electron microscopy, of course, gives us this higher resolution. But for instance, if we look at this endosome here, it has a tube sticking out. There's another one here that is sticking out. From this one static image alone, we will never be able to determine whether that is actually moving away or is fusing with this endosome. And so by bringing light microscopy and electron microscopy together, we really say 1 + 1 is 3, because you get more information than you would get from using both technologies in a separate system, workflow. Now, there have been many workflows have been proposed, and you can put that in words, and you can put in a schematic all the different types of light microscopies you can use, different types of electron microscopy, and how you process in between. But there's one important thing, and that is that the biological question itself should drive the technology. So, and I'll try to explain that with 2 examples. So this is a project where we worked with Jess Carlton from King's College, now at the Crick in London, and he's looking at the escort-free complex. And he noticed that that escort-free complex is present on the reforming nuclear membranes after cell division for about 90 seconds. And so when cells divide, they break down their nuclear membrane, the cells divide, and then when the cells are reforming again, also the nuclear membrane is reformed. And those red dots is the escort-free complex, and that is present for those 90 seconds when the nuclear membranes are reforming, and that is on a cell cycle length of about 22 hours. So that's about 0.1% of the time in the cell cycle of a cell. That complex is present at that specific location. That is less than 1 in 1,000 cells. So if we would just start doing electron microscopy on that, we could search for days to find that particular cell. So we're going to use an aid. And so we have the fluorescence, which tells us, okay, the protein or the protein complex is currently located at that position. And what we're growing our cells in are special dishes. And those dishes have a finder pattern embossed in them. And so we can note, in this case, it's 5Q, and we can note that down. And then when we've done the imaging, we note the location, that pattern is embossed in our plastic resin. And so we can trim down to the right area and trace back our cells. So we can really go from the light microscopy, where we have our protein complex localized, to our overview image. So this is that dark structure is the reforming nuclear complex, nucleus. And then we can look at that, and we've used a trick here. So attached to the fluorescence are nano gold particles. We silver enhance those gold particles when we process for electron microscopy, so they show up as dark particles in the electron microscope. And so now we're going to reconstruct that nuclear membrane. And so here you have the nucleus, you have the cytoplasm, and this is that reforming nuclear membrane. And we've reconstructed that. And so the membrane is in red. The gold particles for that escort-free complex are in yellow. And what we found is that that escort-free complex is present at those very tiny last holes which need to be sealed by that escort-free complex. These are not nuclear pore complexes. We checked for that. And so what we've shown by using that capture of the time of 1.1%, and then using our technology for correlating that, we could really show that that protein complex is present at the sites where the last parts of the membrane are sealed. Now that was relatively simple because we have that finder pattern underneath the cells we're interested in. Now we're going to look at a more difficult system, because in brain, of course, we are 3D structures. And so if we want to look into tissue, we have a massive added problem. How do we find back our cell of interest in a 3D volume? We can't use those cover slips because we can even do the imaging live in awake animals. And here we're using multiphoton microscopy to really do deep imaging. And so this is more like finding the needle in the haystack, because you can see this is a 3D volume. You can see the labeled nuclei. We've also injected one specific neuron we're interested in after fixation. And now is, how do we find back that one particular neuron? If we would, again, just go into electron microscopy, that would take us years to potentially to find back that particular neuron. So what we use is we're going to use the power of one of the lasers for a multiphoton system, and we're going to burn marks around the neuron of interest. And slowly what we're going to do is we're going to make more brands, but we're going to increase them in size. And so we build a pyramid. And now this pyramid really guides us to our neuron of interest. And so when we do that, this is our neuron of interest. And we're going to slowly burn our marks in there. So that's on the left is the pre-brand before we do the branding. And then in the right, you can see the brand marks present. Here they are. And the next one is coming up now. So I hope that's visible. And this is only two, but the tissue higher up also has these brand marks. And in a Z-stack bit cleaned up, that's much easier to show that they are on different heights. They are a bit overlapping so that we can see where the transition to a smaller quadrant is going to be. And then slowly we start making semi-thin sections from the light microscope, and we can see the brand marks in that tissue. And then slowly we're really gonna zoom down until we are in the area. And this is the smallest area. This is now where we want to start looking. Here, we're going to use a different type of electron microscope. We're not going to use a standard transmission electron microscope. We're going to, in this case, use a scanning electron microscope. But they've pulled a trick here, and they've built a microtome, a slicing device, inside the SCM. So I've sped up this movie twice, just so you get a feel. What it does, it has this microtome inside the scanning electron microscope, and it slices off the top, scans the surface, slices off the top, and that continues to do that. So in that way, we can build large volumes of high resolution ultrastructural data. And that's what we've done here as well. So this is the top of our block, and this is one of the cells of interest, and we're now going to build that whole structure. And so these are all slices. It's scanning. And now you can nicely follow. So here's that axon running that came out of that neuron, and we can trace that for a long way. And so we can see that in the light microscope, but now because we have such a large volume and we can do this large volume imaging with that scanning electron microscope, we can follow that neuron and look at it in high resolution. So we can much better look at connections between cells. So this serial block phase SCM, as we call it, is used a lot in the connectomics field. And so in the end, what we get is a large volume of data. And so you can see the nuclei, and we can reconstruct that volume. The parts that are interesting to us, we're going to color in as we do, and then we can look much better at those connections. So that will appear right now. That green one is the neuron we were interested in. And so we can now compare that. Sorry. You can see this extension. So on the left side, before the near infrared branding, we can see the cell body and the two extensions. This is probably the axon that is extending down. We can see the brand mark, so we know for sure that that's the neuron we've been looking at. And then when we've reconstructed that whole volume, you can see that this extension corresponds to this extension, and the axon is extending downward. And so by using this trick of making these brand marks, this pyramid of tracks to our object of interest, we've been able to find this one particular neuron amongst millions of other neurons. And so this was a project that's done by Rob Lees. He was a Wellcome Trust PhD student. He's now moved to Oxford and hopefully continuing with this process. But he set up that whole procedure with guide from others. And so this was a project that we did together with Lucy Collison from the CRIC. And this whole volume imaging is really taking off. So we had the revolution of cryo-electron microscopy. I'm sure most of you have heard about that. There's now a whole movement which looks at molecular detail. But there's this whole other movement that looks at connections, larger volumes, and this VEM community, we've really set up a whole community discussing how we should approach that. And we've had 2 town hall meetings in 2019, 2020, and later this year in October, we'll have a 3rd town hall meeting really to organize the community, see what the needs are, because it's not a completely widespread technology yet, what kind of the developments are needed. And so if you would like to be involved, please subscribe, and you can actively take part in one of the 6 working groups we set up to organize that community. My specialty is correlative light electron microscopy, and I love that technology. And so I really like to spread that technology as well, just look at the strength, but also how you have to think about setting up a CLEM experiment. And so we've gathered a lot of protocols, because as I said, you can do it in tens of different ways. We've accumulated protocols in a series of books, and we organize MBO courses on correlative light electron microscopy in Bristol. So if you're interested in that, have a look or send me a mail and I can send you a link. And to finish off, of course, it's nice that I'm presenting, but the real work is done by a lot of other people. Most of the CLEM work is actually done by Lorna, a very talented postdoc. Most of the electron microscopy is done by Judith. She's been with me in Bristol since I moved there in 2006, so a long-standing collaboration. Chisato, Daniel, and Celia are 3 relatively new PhD students. They've only just started, where Chisato is looking at cryo correlative microscopy, and Daniel and Celia are looking at correlations between light microscopy and, for instance, something like impedance. And so that's a whole new different area. You, Jen, and Rob are PhD students who have left, and Boko is going to join the group later this year to work on electron microscopy. And with that, I would like to finish and thank you for your attention. And I'm, of course, happy to answer any questions.
That's fantastic. Thank you, Paul. Yeah, I've been fascinated with cryofluorescence and CLEM ever since we had that first test at Bristol, where we put our confocal on your microscope just to see what would happen. And yeah, I've been bitten by the bug ever since. So hopefully you'll see a few of the updates that we've done in the next couple of talks. So if anybody has any questions for Paul, please put them in the chat window. I was really impressed by those fantastic images, especially the final render of the volume. What software did you use for that rendering?
Now my mind has gone blank. It's not Aviva. What?
Arevis?
No.
Avia.
It's Thermo Fisher.
Okay.
It's acquired by Thermo Fisher.
Okay.
Although they do remain independent from Bordeaux.
Okay. If you think about it, maybe put it in the chat window, because it's done a really good job. That's for sure. Is there a limit in the depth you can go to doing this volume CLEM with this either serial block or your brand marks? I noticed you were stepping in as you're going down. So does that limit how far you can? Stephanie Ray says it's Imaris.
Yes.
That's it.
And well, we used Amira. Now I remember.
Amira, okay.
The Amira version, which is the same as Imara. Yeah.
Okay. Yeah. So the depth that you're going down, this sort of diagonal stepping down, does that limit your depth?
Yes, that's basically the power of your laser that limits that how far you can go, but you can go pretty deep with the multiphoton. Yeah.
Okay. And I mean, over what time? I always think about time of taking these experiments to do these multiple steps to go in deep. I mean, how long does it take you to get one of those fantastic images?
Oh, so this we can do the, let's say, the imaging itself. We can do it on live animals. So we've traced where we've made a cranial window and imaged first, and then a week later the second time, and then of course euthanized the animal and made the brand marks afterwards. You can also just take brain slices and cultivate those. So that's the branding itself, half a day of work, not even. The processing for microscopy takes a day if you do it all in. Then the trace back, that's probably the most time consuming part, and you don't miss the near infrared branding marks.
Understood.
Because that is the tricky part. And then once you set that up, once you found your area of interest, then it's an automated process. You set up the microscope to take slices every 50 nanometers, and then you let it run overnight or sometimes even over the weekend, depending on the volume you'd like to have. But of course, you're generating massive amount in that case.
Sure. So how are you moving that data around? Do you have a dedicated fiber network or something?
At the moment, we don't have a good system yet. At the moment, it's mainly on hard drives.
Yes, and with our new microscope, that we can create quite a lot of just fluorescence imaging data, and already with like how do we move about and store all this data from demos and things. So yes. Your problem is multiplied somewhat from that.
Yeah.
Okay, thank you, Paul, so much. I hope to see you soon in Bristol,
yes.
when we're allowed to meet again.